Our protocol allows us to visualize how fluorescently tagged cells respond to an inflammatory peripheral stimulus in a live animal in real time. Two-photon imaging allows visualization deep into the tissue of a live specimen, preserving the integrity of their cells and their microenvironment, and providing an accurate representation of the biological system.
Breathing moves the paw over time, causing blurriness and a loss of the focal plane. Be sure to affix the paw firmly with tape to a stable surface before imaging.
Individual may struggle to find a focal plane of interest. Make sure that objective is close to the paw without touching and is directly above the injection site.
Before beginning the procedure, turn on the multi-photon system and select the 25x objective. Place a stereotaxic apparatus onto the stage of the multi-photon microscope and connect the apparatus to an anesthesia delivery machine. Place a piece of matte black paper on the surface of the apparatus as a connection point for the mouse paw. Set the resonance scanner with a fixed scan area of 512 by 512 micrometers. Tune the excitation lasers to the 930 and 1100 nanometer excitation wavelengths for green and red fluorescent protein signals, respectively. Use a dichroic mirror of 690 to 1050 nanometers to direct the light path of both excitation lasers to the single objective, allowing the 930 nanometer tuned excitation laser to be reflected to the main scanner, and the 1100 nanometer tuned laser to pass directly into the main.